Okay, moving on. So, okay, we talked about the limitation in the time step and the fact that we cannot take too long time steps. If you take too long time steps, the system becomes unstable, a variety of reasons and problems. But then, in addition to instability of your simulation, if you want to describe a molecular system, you should have a few snapshots for every vibrational mode of the system. For example, if you look at the molecular system like this, usually the fastest vibrating bonds or modes are bonds. The fastest vibrating modes are bonds. And then among bonds, those bonds that include light atoms are the fastest, namely hydrogen bonds. Bonds involving hydrogens, let me not use hydrogen bonds. Bonds involving hydrogens are the fastest vibrational elements. And their period of vibration is about 10 femtosecond. So they go in 10 femtosecond, they go back and forth, one full period. And in order to describe that motion properly, you need to take 10 snapshots. Otherwise, you don't get the. So if you take one snapshot every 10 femtosecond, you will see that hydrogen always the same place, right? If you take even actually every 5 femtosecond, you might see it because there is a vibration periodic function, you might see it always at the same point. So it's good to take about 10 snapshots for each motion, and because the fastest degree of freedom you have here is a CH bond like this, and that sort of vibrates in a 10 femtosecond period. 1 femtosecond is a good choice. So now, if you look at, again, what happens in biology, so this is our time step, 10 to the here, time step, 1 femtosecond. So bond stretching is around here, as I pointed out, 10 to 100 femtosecond. Then in a picosecond regime, you start looking at sidechain rotation on the surface of the protein. Then when you come to the nanosecond kind of time scale, then you look actually hinge bending, microsecond. Excuse me. And then most of the sort of relevant biological phenomena happen on a microsecond or longer time step. So these are the numbers you see here are a kind of approximation. If I can do 1 nanosecond of simulation every day, then in order to do a microsecond simulation, it would take a year or 3 years to be able to do that. But nowadays, so you can easily go to 20, 50, even 100 nanosecond of simulation every day. So we are reaching nicely here. If you use a specialized hardware like Anton, you can easily do many microseconds every day, so you will be more in this regime. But the point is really a lot of interesting biologically relevant phenomena are in this regime. So we do whatever we can to increase the time scale of our simulation. So one way is, for example, because these hydrogens are very light and they are moving too fast, so we can somehow have a different representation of the molecular system and get rid of the hydrogens. For example, you can say, I'm going to include these hydrogens into their heavy atoms and just have this carbon weighing 3 units more, this carbon 1, this oxygen doesn't have to worry about. So by removing the hydrogens, you can actually now get rid of the fastest degree of freedom, and maybe you can go to 2 or 3 femtosecond time steps. Or if you say, okay, I'm going to prevent them from too much stretching by restraining them. There are algorithms that constrain bonds, including hydrogen, Radel, Shake. These are some algorithms that are implemented in the program. And therefore, when you do that, then you don't have to worry about this very fast vibrating mode. you can easily go from 1 femtosecond to 2 femtosecond, double the time scale that you cover with the same compute. There are really some overhead costs by including these algorithms, but in total you will gain some. So there are other methods also to try to accelerate the system. This is the beginning of coarse graining the system. So instead of having hydrogen explicitly, we can bag them together in one particle and make it a sort of coarse grain model. But you can do it at a higher coarseness. So let's say, what about if I just have for the whole backbone here, I will have one particle. For the whole side chain, I will have one particle. Now you have 2 particles instead of 10 or 15. Plus, you don't have all these light particles that give you these very fast vibrating modes. So by doing that, you will decrease the number of particles, and you can also take much longer time steps. Instead of 1 femtosecond, you can now go to 20 femtosecond time steps. So that way, the time scale increases significantly, but of course, you lose the chemical details. So you don't have this hydrogen bond anymore. So that's the trade-off. But anyway, so now I think we talked about this already on the first slide I had. So again, initial coordinates might be bad, high energy. You minimize them, cool them, and then you thermalize the system. You start crossing the barriers. And here in the first basin, you found in this picture, you found the global minimum. And you sample, because this is where the system, in reality, when you do experiment, the system will spend most of its time here. These are too high energy compared to here, KT, 2 KT, 3 KT. They become exponentially less probable and contributing less to the average property that you're measuring experimentally. So you want to sample this as much as possible. But again, the landscape might look like this. So what you have, in order to really get the real behavior of the system, you have to be able to sample all of these. And depending on how high the energy is, becomes a time scale problem. This is one example of sidechain rotation. So if you monitor, for example, this is a simulation, 1,000 picosecond or 1 nanosecond, and we are looking at this dihedral angle formed by these 4 residues describing chi1, chi2 rotation of this tyrosine side chain here. And you can see, if you look at just this part of the simulation, you might think that, okay, we are equilibrated here, so nice, everything is stable. This dihedral angle is in this particular conformation, fluctuating a little bit, of course, but not doing anything. But as you run longer simulation, you start seeing that it flips into a different configuration. So now, let's say, now what am I going to do? Is this equilibrated or not? So then depends on my definition of a state. So if this conformation is one state, I can define this as one state, that one as a different state. For this state, I think I have, for this basin, I have an equilibrium system, sort of it is fluctuating. But if I want to consider both states in my ensemble, this system is not equilibrated. You need to run this long enough to see back and forth transition between these 2 states, enough back and forth so you can claim that the system has had enough time to go from one basin to the other, and then the probabilities of correct. And you can even compare the probabilities to say something about the free energy difference between these 2 states. So right now, I don't know if this was a bad initial state, just clicked into the correct conformation, and that's going to go forever, or is it coming back later? So I can't say based on this. And if sometimes I have seen some people trying to say, if I look at my simulation, now 80% of the time is in this state, 20% of the time is in that state, and then therefore the free energy difference of this should be a lower energy state, this should be higher energy, but as you can imagine, if you run this longer, you might have a different probability distribution. So you should be careful. Even if it sort of stays there, once you come back, you don't have enough transition between the two states to say something. You see, I mean, you have some little transition here. You have to define your state, what is up, what is down. So really defining the states, defining the reaction coordinate is something that you should do, and it can be non-trivial in many cases. Actually, we talked about this already, so that's a repeated slide. So another thing, actually, another step that we do quite often is the solvation. So you might wonder why solvating the system increases the size of the system, the number of particles increase in the system because you add solvent molecules. But I think it has been probably the most common way of simulating a lot of biological systems, especially since dealing with larger molecular systems is easier on parallel computers. If you have a lot of nodes, your simulation scales much better. So, and if you don't have the solvent, of course, again, it depends on the level of details. If you have a very coarse-grained system, maybe you don't care too much about explicit solvent molecules. You can have implicit solvent molecules in the molecular system, in your simulation system. But if you are dealing with, heavily charged systems, like DNA, ions are important. Obviously, solvent becomes important. So we have a lot of implicit solvent models available, but they have become less popular because there are lots of approximations in them that are difficult, like what is the dielectric constant of different regions of the system, and how do you define the regions, what is protein, what is membrane, what is solution. These are not trivial. It becomes very complicated. And as I pointed out, adding solvent is not too expensive, especially on supercomputers and clusters of computers. So that's why explicit water and solvent has become the more common representation. So there are implicit solvents. I mean, if you have a huge system in which it makes a huge difference to replace explicit water with implicit, then you can do that. Sometimes you can have hybrid systems. Let's say I would like to have a little shell of water around my protein just to make sure that the protein is not completely naked and I don't have crazy electrostatic interactions on the surface, but then the rest of the box can be treated using a continuum description or implicit treatment. In some cases, we have membranes. So and again, this is also something that increases the complexity and the size of the system. But again, lipids have been sort of shown to play really important roles. It's not just a low dielectric environment for your protein. We started to learn that they are playing explicit roles. I showed you the movie about the ion permeation through the space between lipids and proteins. So they are playing roles, and it's just a matter of time for us to learn that they are playing role in most membrane proteins probably. There are specific lipids that interact directly with the protein, participating in the mechanism. Sometimes they are almost part of the protein structure. There are some mitochondrial proteins that are so tightly bound to cardiolipin, you have to boil the protein to get them separated, and they don't work if you don't have enough cardiolipin. So the lipids are almost part of the protein structure. So they are playing important roles, and this would be really the most realistic representation of the system. So I talked about this again. So the initial positions come from PDB, or you put together a model using Charm GUI or your own script, Solvate in VMD. But you also need to have initial velocities. And for that, so we are using a probability distribution of velocities in a way that it matches the temperature that we want to simulate. And this comes from the, excuse me, sort of Maxwell distribution of atomic velocities, in which your temperature here is included, and that's by adjusting the temperature, you can get the kind of distribution that you would like. And from this probability distribution, then you randomly assign these velocities to the atoms in the molecular system. This has to be done only in the beginning of the simulation, because then at a later time, every time you are calculating your velocity based on the initial velocity and the acceleration. So these files are saved in NAMD runs. You can tell the program to save these files every so often. So you specify that. Of course, saving them too often slows down. You don't want to do that. But saving them, I mean, I don't know, about 1,000 steps or so might not be a bad idea. These are the restart files. You save the coordinates and the velocities, and these are the two things that you need to continue the simulation. So oftentimes you can only run for a few hours, for 1 day, 2 days on the computer queue or the supercomputer that you have access to. So it's good to have these restart files so you can continue the same simulation with the same exact velocities and positions. Yeah.
You get the same position as a nanosecond out if you use the same starting files?
So there is, again, I mean, there are not exactly the same. So there are a couple of things. So that we have some random terms coming in that shake things. And then also there are, at the level of sort of hardware and rounding, there could be some errors there. But for the most part, I think actually these are, except for the noise term in the Langevin part, these are pretty deterministic. So you can, pretty similar. So if you run it longer, then again, the random term can start affecting. But you can also control that, like you can specify the random seed number initially. If you save that one, that random number that initially is used is exactly the same in two simulations, then you get pretty much the same trajectory. That's right. Okay. Okay, so then you run the simulations. Again, I think you can set up the simulations today. You have the PDB that includes the coordinates. You generate a PSF that has the topology and the atom types. We will talk about this tomorrow in more detail. And then you run a NAMD simulation. So in your tutorial, you have pre-computed trajectories from your simulation. Then you start to analyze the system. The system we are using is ubiquitin. Ubiquitin in water box or in water sphere. There are some different examples there. And one of the most basic analysis we do is RMSD. So this is root mean square of displacement or deviation. So in general, so you see, I mean, the main point is you are dealing with a messy system. You look at it, is it a square, is it a sphere, is it a rod? So you have a bunch of atoms and you want to somehow analyze this. So going after position of individual atoms might not be a good way to start with. You want to somehow come up with some what we call collective variables, coarse coordinates that describe the shape of the system, the motion of the system, the behavior of the system in a more collective manner. If you have a helix, for example, you can define a vector for the helix, for the whole helix, and see what the angle of that vector might be during your simulation. So RMSD is probably the most global collective variable, because here you can include the position of all the atoms. So I have this thing was here, and now it's here. What is the RMSD? How much it changed after I started my simulation? So it might have translation rotation. During the simulation, you remove those by superimposing the initial and final. And then you look at how much it internally changed during your simulation. So that's the easiest way to look at the average displacement of individual atoms. It could be just the backbone. It could be just the C alpha atoms. It could be all the heavy atoms. You can include all these things in your RMSD analysis and see how it changed. So this is the RMSD for the entire system. You can see that initially you always get something. If you're looking at 100 residue or so, you always get something at least 1 angstrom. That's part of the fluctuation of the system. Even if you start with an equilibrated system and you continue that simulation, there is always this much fluctuation, 1 angstrom spread of conformational fluctuation. And then after that, it sort of starts to plateau. So this you could say, based on my RMSD analysis in a 1 nanosecond simulation, it seems that this guy has reached an equilibrium. It's not going farther. Again, you might run this, and I don't know, 10 nanoseconds later, you might have another surge. Something happens to the protein, but then you can consider that a transition to a different conformational state, maybe. So you can, RMSD is good, but then you can actually do this differently. So when you do RMSD, you superimpose, again, your protein might start here, shaking, and then at the same time moving. You superimpose the entire protein on the initial conformation and see how much it internally changed. So now you might want to see maybe that doesn't quite capture the motion between the domains of the protein. Like, let's say initially I was like this, at the end I was like that. If I superimpose these two, I see the global change average over the entire protein structure. But maybe this motion was mostly about this might be a better example. Maybe I have a flap that closes and opens. Most of the protein is not going anywhere, doing anything, but this flap is doing it. But by just superimposing the whole thing and looking at the average RMSD over the entire thing, that doesn't quite give me how much this part is moving. So what I can do is, let's say, first I do general RMSD, then here you can put the RMSD values for individual residues, right? You can see how much every residue is moving, and put the RMSD value in your PDB file. This is one exercise you go through this afternoon. And then load it in VMD and color the protein based on RMSD. So now the blue parts are low RMSD regions. The red parts are high RMSD regions. So now next time, I understand, I see that there is a core part of the protein that is quite stable. I can use that as a reference for my superposition, for my alignment. So now I use all the trajectory, use this core part, superimpose them based on this, and then I can capture this motion much better for my paper. So RMSD of this domain with respect to the core domain was 5 angstroms. Whereas if you do the entire RMSD, you might get 1 angstrom because it's diluted. So that way you can actually present what you think is happening much better. So for the analysis part, there is a lot of information that you can just simply see by just looking at the system. That's a very powerful way of analyzing the system. And it's not just about making nice movies and nice figures. Your visual cortex is so powerful in seeing a lot of things. And these are very complex systems. It's impossible to script everything. So you watch the simulation in VMD, you watch your trajectory, you might say, oh, I've been always interested in this part I'm analyzing, but maybe something is happening here that you have been missing. By just looking at it, turning it around and watching your trajectory, you might notice something quickly. And then, but that's not enough, because then that might be human error. Then you have to quantify that. So you find this flap, and then you put a graph there that shows what is happening. Then you put together an initial conformation, final conformation, and a data set together in one nice figure for your paper. If you just put that one there, maybe I read your paper, I have no idea what that part of the protein is. What does it mean? Oh, there is a part in the protein that is moving, but what is it? Where is it? It doesn't tell me enough information. So the combination of a sort of graphics, molecular images, and data proving that the phenomenon, what you think you have found is real, is very powerful here. Okay, so you can, RMSD is the most global, but then you can also define these collective variables for different portions. So let's look at this helix. Does it remain helical? I can have a helical property of this quantified in VMD and plot it over time, show it's stable or it bends. What is the orientation of the helix with respect to the core of the protein? In my transporter system, what's the orientation of this? So I can come up with different sets of collective variables to describe this very complex 3N system in a more human understandable manner. That's the art part. So that's what I was telling you. So you can get the RMSDs per residue. There are utilities in VMD that allows you to do that, and then color your protein based on this. An example of this is potassium channel. So now if I look at the I can look at the entire protein RMSD, which is the whole thing. Also looking at the selectivity filter separately to see if something is happening to the selectivity filter separately. You know, you can distinguish between these different types of RMSDs and what they might mean. And again, this is an example in which we are looking at residues, per residue RMSD. So the helical parts. What are the helical parts? The N and C termini, so these are flexible regions. They move a lot. And then there is a loop in the middle that also moves more. So everything else is pretty stable, below 1 angstrom RMSD, which means all the helices and the selectivity filter are pretty stable. Okay, so now, so I decided the second part I would like to talk about a few ways that we have been trying to battle the time scale fight. So there are, I'm going to talk about two different classes. One is doing non-equilibrium MD simulation instead of waiting here for the system to go back and forth billions of times and maybe crossing this. Once we can actually help the system, so along this reaction coordinate, and that's actually another thing that you have to do it correctly. So you have to define a reaction coordinate. What is it? Is it the distance? Is it the position of the ion? Is it the orientation of a helix? How you would like to promote or excite a particular mode in your molecular system. You have to decide about that. You have to design that. But once you have that, then there are ways of encouraging the system to move a little bit more in this direction. So you will have more biased motion in this direction that helps it to cross this barrier. So these are non-equilibrium systems or biased simulations. And then we have also reduced representations in which, first of all, this coarseness is going to go away. You are going to have much shallower energy wells. These barriers are going to be lower. And you're going to have less particles, less number of particles that allows you actually to do calculations faster. So let's look at these two different classes, reduced representations and non-equilibrium MD simulation. I give you some examples. There are many different methodologies in each class, but I try to give you some examples. One very common sort of very useful protocol or trick that has been used for a long time. It's called differently in different programs. SMD is one of the most common names. Steered MD, you steered your molecular dynamic simulation in a particular way. In addition to the forces that you get from your force field, from your bonded terms, from your electrostatic interaction, in addition to those, you add external forces to the system, depending on how you want to steer the system. So I am here, let's say I am an ion, and this is a channel. I would like to, and that's I want to go through that door. So that I have to go through this pathway here and exit the room. And you are just watching me here. I'm doing equilibrium simulation happily because I'm blind. I don't see where I'm going. I just hit different particles, feel different forces, and you might watch me here for weeks just sampling this space. But you really like me to leave the room because you want to go to lunch. So what you can do is to maybe, if you know that's the exit, then you can add a little extra force on me while I'm still doing my fluctuation. Then I'm encouraged to move more in this direction. So instead of waiting for pure diffusive behavior and thermal fluctuation to get me there, which it will at some point, next year maybe, and you don't have time to watch it, so you can actually encourage that. So that's the idea behind the steered MD simulation. Now, the force can be applied on different atoms or groups of atoms in different directions. By combining them, you can create different types of biases. So for example, I don't have any examples here. So for example, if simple ion permeation or diffusion can be done by applying a force to heavy atoms of your substrate. But you can also, let's say there are two proteins and you want them to rotate with respect to each other, so you can apply tangential forces to all these atoms and create a torque in the system. You can, what we have done in the past is, for example, to have a membrane in the middle and all the water molecules above, all the water molecules are pushed in one direction, the membrane is fixed in its place. So therefore you create a positive pressure on the top and negative pressure at the bottom. That gives you a gradient for the hydrostatic pressure. That's how we created more water moving through the water channel, for example. So you can use them to create a bias. So now, the problem here is, of course, first of all, you don't know what is my natural time scale to leave. If you know from the beginning, so if it takes a year or it takes a month for me to exit the door, that would be useful to sort of gauge how much force you need to apply. But oftentimes, we really don't know. We just know that, okay, I did 10 nanosecond simulation, or I watched a mod here for a day or two, still here. I don't know what's going on. So that's, we don't know what we should do. So what we do is, let's see, I have slides on constant. Oh yeah, I have, okay. Okay. Yes, the problem is, okay, so oftentimes you have to apply the force. So now, if your force too high, so then instead of taking the natural low energy pathway, which is here, then I might actually go over these chairs, right, over these tables here. So that gives you high energy kind of barriers because you're taking an artificial pathway for me. Then the mechanism is incorrect, the pathway is incorrect, and the energetics along this pathway is incorrect. So that's the problem. You can cause hysteresis because you don't give the system enough time to explore the space and find the easiest way of doing things. So that's the problem. But still, there is a formalism allowing you to connect the work that you are doing to the system when you are adding force and you have displacement. You are working to this. You are adding work to the system. And based on the second law of thermodynamics, your work in a macroscopic system is always larger or equal the free energy difference, right? So that's the second law. So every time you work on the system, if you do it very, 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 very slowly, so the work is all reversible, you get your free energy difference. If you do it faster, then you get non-equilibrium into non-equilibrium part, and you lose some of the work. Your work that is done to the system goes partly to heat and dissipates, and you lose it. Your work will be higher than free energy. But in a microscopic system, in a molecular simulation or a single molecule experiment that you do, things are different. So this is the scenario. So you start with an initial state, and then you go to a final state. And during this is you exchange work and heat, and you are interested in the delta G between these two states. So here is the situation that we have in a microscopic system. If you look at the distribution of the work, you get a Gaussian distribution of this sort, okay? And the average of the, and here is your free energy, the actual free energy change as you go from initial to final state. And if you look at this Gaussian, there are ways to show that this distribution, the average of work according to this distribution is going to be higher than the free energy difference. That is what you're interested in. But if you look at this distribution, you see that there is a little tail here. There are a few experiments. Let's say these are all ions going out of the room. So every time you're pulling me, you're spending too much effort to get me out of the room. But there are a couple of cases in which I'm going by myself out of the room. My fluctuation, everything is in the correct direction. You don't even have to do anything to. You just let me go. That would be your best bet. So in those cases, the work you are doing to the system is going to be less than the free energy change, because most of it is coming from thermal fluctuation at the microscopic level. So this little tail, still the majority of the experiments or the pulling simulations are using too much work compared to the delta G. But there are rare cases, a few cases, in which your work is going to be smaller than free energy difference. So instead of looking at the work value, if you look at the exponential distribution, then there is a match between the exponential of the work average, exponential average, and the free energy exactly. So there is a formalism showing that this equality exists. And this is because this negative exponential minus beta W is dominated, it's a negative exponent. Therefore, it's dominated by small values of W, meaning these guys. So these guys are, the W's are small, then this exponential becomes a larger number, and they dominate this average. So if you run, if you repeat this simulation, or if you repeat this experiment enough to sample this part of the distribution, then this formalism allows you to get exactly the free energy difference, which is your ultimate goal, to describe the free energy change of the process. That's usually the goal. So you can get that from non-equilibrium processes. So this was theoretically shown in a series of papers. These are the two last ones. Sometimes it's referred to as Jarzynski's identity. It was also experimentally proven by Bustamante's group. They pulled a stretch of nucleic acid DNA, I think, experimentally using single molecules. And they showed that you can get the free energy within 0.5 kilocalorie per mole for that unfolding process from non-equilibrium. They did a lot of fast pulling, and then they had one very slow pulling that gives you the actual free energy, and then you can show that. But they repeated this hundreds or 500 times. So now in simulations, you can do, if you can do this process many, many times, then you have the formalism in place to get the free energy. But again, as you see, so this is only possible from many, many, many repeated non-equilibrium simulations. So then again, this many, many, many is you're changing the sampling problem from one form to another. So instead of doing this, watching me for a year, you probably have to watch me in a biased manner for maybe 1,000 days or something like that. The sampling issue is sort of still there. It provides us with a formalism to do things a little bit better, but I think the sampling issue is still there. So when can we use, why are we talking about this? Again, lots of processes cannot be simulated. You cannot watch me for a year to leave the room here. So what can you do? Either you can give up and say, I'm not going to study this process. There's nothing I can do. It's too slow. Or you can say, OK, so let's look at the biased simulation. Let's promote, encourage them out to leave the room, and then see what happens. And based on that, we might get some actually idea about the path. I might indeed, even though you are pulling, I might indeed take the right pathway. So you have at least a little bit better sampling of the space instead of just watching me in front of the podium here. So you sample a little bit more of the space. You can maybe start one of the systems. I will be over there. You start a simulation there, do some equilibrium simulation, and sample a little bit better. So you have a chance to explore the system better. Now, this is just one example comparing two water channels. One of them is a pure water channel, only allowing water to go. The other one is a aqua glyceroporine, meaning that it's, in addition to water, it also allows glycerol to go through the channel. So we wanted to compare these two systems. In the case of water permeation, that was easy. It just happened in a few nanoseconds because water, you have a lot of water molecules in the system. What's the concentration of water? I'm just buying some time to drink some water. What? 55 molar. And what's the concentration of most solutes in biology? 100 millimolar. So there is a, what is it? 550 times more water there is in the system. So you have a lot of water molecules. It's small. It just goes through the channel on its own. But for glycerol, even if you go very high concentration, so you might reach hundreds of millimoles, and it's much larger, and it has a hard time to go through the channel. So we see water permeation on its own. I showed you the movie. But for glycerol, it doesn't happen. So what can we do? We decided to do glycerol permeation through a glycerol channel. Just pull it through the system. And we decided to also repeat it for this channel. We know it shouldn't take place, but we would like to induce this artificial process and see if we can compare the energetics of the two processes. That's one way of finding out where are the high barrier regions, where are the constriction regions. Is it electrostatic? Is it mechanical? What is it that allows this channel to be a glycerol channel, but the other one doesn't? I already have the answer here for you, but so this is, by the way, for those of you who are looking at channels, these are representations created by the program HOLE, H-O-L-E, that places the spheres according to a particular algorithm, and then at the end it gives you this sort of bunch of spheres. You can load it into VMD. It also gives you the radius profile, telling you how wide your pore is at any particular Z. So we did the simulation, and now I'm talking about the different protocols for SMD, state MD. You have to apply a force. So now, should I apply a force of 10 piconewton on a mouse, or 100, or 1,000 piconewton to make sure that he leaves the room before lunch? That's your limited time scale that you can simulate. I can simulate my system 100 nanoseconds. This is my allocation on my cluster. It takes me a week. I know that from my benchmark. So what should I do to make sure that my event takes place in 100 nanoseconds? So if you want to use a constant force, you never know because you don't know. You have no idea about how slow this process is or how large the barrier down the road is. You don't know. So the constant force, and we have non-constant force, 250 piconewton in this case, for example, and it worked for this case. You can see how it behaves. This is the time versus displacement. And you can see it just sort of moves to here, gets stuck a little bit here. There is another barrier here, a large barrier here. It spends a lot of time to try to cross that barrier. So every point it feels the same force. It might be too large. Maybe you didn't have to apply 250 piconewton. Maybe I would go with 20 piconewton, so which would be more natural, less artificial. So that's a little bit difficult. You might do it for other purposes. The better protocol is what we call constant velocity. This is constant force. This one we call constant velocity, or CVSMD, in which I would like to look at it as a variable force. This is constant force. This is variable force. I would like to have a protocol in which I slowly increase the force higher, 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 until the system complies or the system follows. So how can I do that? So instead of pulling them out directly, you can connect a little spring to me with a particular force constant and then pull the other end of the spring at a constant velocity. So if I'm not following, you keep stretching the spring, increasing the force acting on me. But if I'm following, you don't, right? And as soon as I cross a barrier and I come close to your pulling point, spring shrinks and your force goes down. So in a very nice manner, with a little trick, by using this spring, you can really adjust the force very nicely. So you just increase it as much as necessary. Once you reach a good force for me to cross the first barrier, I start to follow, and the force drops. So this is the most common way of applying SMD in NAMD. Plus, you can actually adjust. OK, I have 100 nanosecond, and from here to there is 30 angstroms. So, and if I have a pretty stiff spring, making sure that Ahmad keeps close to the other end of the spring, you can assume that I will be following closely the pulled end of the spring. Then you can calculate, OK, 30 angstrom over 100 nanosecond. This is how much displacement I want to have. So you can calculate the velocity. In this case, this is super fast, 30 angstrom per nanosecond. This is not too large, but still, I would say for current standards, it's too fast. So 30 angstrom per 100 nanosecond would be like 3 angstrom per 10 nanosecond, or 0.3 angstrom per nanosecond would be the calculations, the speed of velocity. Yes?
Do you pull from like the other side of the membrane? You allow it to choose its own path, or do you have to pull it locally and you determine the path as you pull?
That's a very good question. So in this case, we are lucky because even though it's a little tilted, the pathway, but so more or less, the largest component is the membrane normal. So we just define a force along Z, the membrane normal, and it's free to move in XY direction. If you don't do it too fast, I'm still free to do and find my way, follow the pathway more or less. But then you might have actually a pathway that is curved linear. It's binding of a drug to a binding pocket. Maybe it has to go this way. That becomes actually really difficult. What is the direction of the force I should apply? So people have tried to come up with sort of smart ways of defining, maybe trying a few different ways and see which way is opener to follow that. So there are ways of trying to come up with a better pathway or better pooling protocol, but it's non-trivial. So you need to have some idea. You have to design it yourself. Yes. OK, constant velocity. And again, I think, so one thing I would like to emphasize here is it's very useful if you have multiple simulations, right? So for a long time, and I think this is still true, if you're doing a mega large simulation, one simulation would be still acceptable. But for the majority of the system, a single simulation is not enough. So these are microscopic processes are stochastic because of all the noise and everything. So you might see something in one simulation that doesn't happen at all in 9 other simulations. So it's very useful to repeat the simulations at least once to make sure that it is reproducible. People have done this experimentally for a long time. You need to have N, to make sure that this wasn't a random process taking place in one simulation. So that's one thing to keep in mind, especially if you are doing non-equilibrium simulations like of this sort. And you are applying additional forces, you're introducing a lot of hysteresis, I repeat. So you might get me over these tables, and then so here is the pathway of permeation. If you pull hard enough, this glycerol can go through this space between the helices out. If you push hard enough, of course, it's soft matter, it opens up and you go through. But this is not the correct pathway for you. So it's very important to repeat simulations, even if it's equilibrium simulations, to have 2 copies, 3 copies. If you can afford 4 or 5 copies, that would be nice. I think the time of single simulation papers is long gone, so you have to have more. So that's for sure. But sometimes you observe something interesting in one simulation. It might not repeat in other simulations, but you really feel that it's right because you can explain it, you can understand it, it fits the mechanism. Don't discard it completely because it didn't happen in other simulations. You can still sort of, if you understand it, if you can substantiate that and connect it to the mechanism, you can still report it. You have done 5 simulations, it didn't repeat, you have done your part, but still you think that might be actually an interesting phenomenon. I think I wouldn't discard it because again, because of a stochasticity, so a process on the average, let's say, takes place in 100 nanoseconds. We say the average time scale of residue flipping is 100 nanoseconds, or domain flipping 100 nanoseconds. But if you do simulations, a large number of simulations, there is a possibility that in some simulation it takes place in 1 nanosecond. There is a small tail of the distribution in which this happens because you have the right direction of the movement of the right atom, this just flips. So in other simulations, it might take a microsecond. On the average, you have it 100 nanoseconds. That's the time scale for the process. That's what you can characterize experimentally, probably. But it could happen much, much faster. So it can. You might be lucky in your second simulation, something interesting happens. Try to reproduce it, but even if you can't reproduce it, don't completely discard it if it's mechanistically relevant. Maybe then you can bias it. Maybe then you can try to reproduce the same phenomena using biased simulations and maybe look at the energies, free energies. So you might be in the, let me go back to my landscape if I can. Yeah, so you might be, let's say, so this might be a second state. So here, let's say we have the 2-state system, and for the most of the simulations you do, you're staying here, seeing nothing, but then you are lucky in one of the simulations, not simulation 1 or simulation number 3, it just happened that you had the right momenta for everything to just cross this barrier and come here, but it never happened again. So now, okay, I did it 5 times, I can get it again, but now I can use bias simulations or even free energy calculations. And if you do free energy calculations, when enhanced sampling, you force the system to visit here. You keep it here. Keep it here. This is umbrella sampling I'm talking. You put an umbrella, bias it to stay here. Then you move your umbrella next. Make sure that it samples this region. Make sure that it samples this region, and so on and so forth. By having a series of umbrellas, you make sure that your system along the reaction coordinate has sampled all the points. So then you can have you can then discount for this external bias that you're applying and calculate the underlying free energy for each piece and then put them together to get the entire free energy profile. So when you do that, then you say, oh yeah, there is another basin here. There is another state there that wasn't easy to access. Only by luck, one of my simulations got there. But when I do my free energy calculations, then I can show that it exists, the energy is low, etc. So you might discover or stumble on very interesting parts of your space in one of the simulations. Don't always just discard it because it didn't reproduce or it didn't repeat, especially if it's interesting for your system. Okay. Okay, so, okay, I really need to speed up. Okay, so we do SMD, we pull the system. You can, again, go in and see how the substrate is interacting, what hydrogen bonds the substrate is making with its environment as it goes through the channel, something to say about the mechanism, about the importance of sidechains. Then this is what we did, you know, based on the formalism of Jarzynski's identity. The sampling here is very limited. I have to declare from the beginning for only 4 trajectories. But in principle, based on that formalism, we can calculate how much force we are applying and this constant velocity pulling. And the force constant we are applying, we can get the force and then integrate the force to get how much work we are doing to the system to push it through. And then based on that, we can calculate the free energy profile. And I guess we were just really lucky that the free energy profile is, if you look at the barriers, they compare very nicely, at least the order of magnitude, to experiments, to, I should say, very rough estimates for experimental estimates of the barrier against permeation of glycerol through this channel. So yes, you can get the free energy. Let me skip the asymmetric profile. And now we repeated the same simulation for the channel that is not supposed to be glycerol channel. So now the red part is the free energy profile for a glycerol going through a channel that is a glycerol channel. So you have free energy barriers of about 5, 6, 7. It's a slow process, but it takes place. If you repeat that for a non-conducting channel, then you get energy barriers that are 20-some kilocalorie per mole. And if you calculate the probability, this essentially doesn't take place. And now you can go in and see where this barrier is located and what are the structural elements that cause those barriers, so selectivity filter, NPA motifs, and they happen to be the narrowest part of the channel. So you can explain the mechanism here. I think they are shown here. The selectivity filter is very narrow, NPA is very narrow, and this is the third location of the third barrier in the profile. So, okay, so that was one example of a very simple kind of biasing simulation. So now that was just pulling the center of mass of glycerol. We try to distribute the force among all the heavy atoms. We usually don't pull hydrogens, by the way. They're too light. If you pull them, they do crazy things. So we concentrate on heavy atoms. We did different simulations. So let's say these are the glycerol heavy atoms. In some case, we just pulled on one of the atoms because we wanted to see if stereoselectivity makes a difference, if it goes this way in or that way in. So you can design your protocol to look at different aspects. So here is another example of systems in which we needed to do bias simulations. So these are membrane transporters. In contrast to channels, in a channel you just open one gate and things go down the gradient from one side of the membrane to the other. Membrane transporters, the majority of these guys pump stuff against the gradient, against the electrochemical gradient, which means they cannot be open to both sides of the membrane at the same time. So they usually have two gates. So one of them is closed. You recruit the substrate from down below. You close one gate, and then you open the other one. So they have to be coordinated in a manner that both of them are not open at the same time. So there is a lot of conformational change involved in their mechanism. That's why they are much, much slower, several orders of magnitude slower compared to channels. This was the distinction before we have the structures for these guys. Anything that was very slow, transport protein, it was a transporter. Anything that was fast, they call it a channel. Until now, lately, we understand that what are the structural basis for these things. So the point being, they undergo large conformational changes in order to pump the substrate. Very important in trafficking of material in the body, both natural and also for pharmacokinetic of drugs. I already mentioned P-glycoprotein as an example of a transporter that we would like to be able to inhibit if possible. So there is this alternating access mechanism. I told you they have to open to one side, recruit the substrate, co-transported ions, and then close this gate before they start to open the other gate and release the material. So lots of conformational changes involved here, and these are just some examples of the type of conformational changes that brings the alternating access mechanism. You know, for example, this one is glutamate transporter that works by the elevator type. I mean, this whole thing is one protein, substrate and ions bind here, and then the yellow domain moves with respect to the white domain up. So now it's exposed to the other side, and as it comes back in, they are exposed to this side. And all these motions are controlled by binding unbinding events. So there is a chemical coupling between chemistry inside the binding site and whether or not they should be able to undergo these conformational changes. So we have to, if you really want to study these, you have to do atomistic simulations. This one is a simple flip-flop, it's an antiporter. This is a symporter. That's another amazing aspect. How come in one case things go together, in one case you have to be partially bound to be able to flip. So that's, again, speaks to the importance of coupling, or how close the coupling of chemistry inside the binding pocket and global conformational changes is. So for those who are interested, we have published a series of papers talking about these advanced protocols, again, defining a collective variable, and that's our reaction coordinate. How you want the system to evolve, in what direction. You want me to go this way, you want me to go that way, you want me to roll, you want me to just shift. So this is what you should design, based on your knowledge of the structure and the collective variables that are available. So we have collective variables that allow you to look at the orientation of individual elements. We have collective variables that allow you to look at the radius of gyration of parts of the protein. You can, of course, look at the distance between the two domains. You can look at the angle between these two domains by defining these collective variables. So you can design how you want to manipulate the system. And then we are showing that actually again, so your initial guess might, let's say you want to go from one state to the other state. Maybe your initial guess is not good. You can do a series of quick simulations. Pull me quickly in this direction. Maybe I go this way, or maybe I go that way, or maybe I go up and come down. You can try different protocols and see which one looks more promising by just looking how much work it takes. So these are the novel aspects of these protocols. You get all the slides in PDF later. But by playing with the protocol and optimizing it using a lot of quick simulations, let's say this whole process might take a millisecond. I can't do, but I can do a lot of 10 nanosecond simulation, trying different protocols, and then see which one takes less work even though a large portion of that work is dissipative and it's going away, it's non-equilibrium, still it tells me which one is easier in terms of protein mechanics, which one, through which protocol the protein is more compliant. And then I can take that one for free energy calculations, for example, by umbrella sampling or other free energy methods. So we have done a series of things. And here is sort of using the Rockies as an energy landscape. I once gave this talk at Telluride. They have nice meetings there. So Telluride is let's say one of the states, low energy. This is a valley. And there is mountain village here. This is another valley. So if you want to drive, there is a road that you have to take this way, go around this peak. So this is the actual optimal transition path if you want to go from this state to that state. But your problem is that you're standing there when you're doing the simulation only having these two states from PDB. You're blind to the landscape. It's at night. You just know with your compass that the other state is somewhere there, but you don't know what path you need to take to go there. So that's why it's good to explore the system a little bit. Usually what people do, which is well defined, is to do targeted MD. So I have to go in that direction. That's my target. So I define the RMSD between initial and final state, and then start biasing simulations in which I worked with RMSD. Very global collective variable, but useless because you're just saying everything. Everything from here should go there. So it doesn't have any specific to it. So that's why it usually takes much more work to push the system, and you have a higher probability to come up with very artificial pathways, transition pathways. But it's well defined, it's RMSD. Every program has it. If you put it in your paper, people know what it is. So you start with targeted MD, slowly decreasing the RMSD toward your target in a biasing simulation. You decide how fast or how slow you can afford this, 100 nanosecond, 10 nanosecond. I want to do it 5 times. Each time 20 nanosecond, that's how much computational resources I have, okay. 20 nanosecond from here to here. Again, you define your speed, how fast your target should move to complete this transition, and you get some initial guess. Then you can take all these points and sort of refine them, relax them, and you can come up with a refined pathway. But if your transition pathway is too far from your initial near linear path, then there is no way you can get that. So what we are suggesting is, again, to do many sampling using different mechanisms. You can think about this cannot be comprehensive. So we do hundreds of different protocols usually, but still people complain, oh, how do you know it's enough? Well, I mean, it's better than one, right? At least. But if you have one, nobody complains. Okay, one RMSD, good. If you do 200, oh, 200 is not enough. Maybe you should do thousands. Because again, it becomes arbitrary, right? I mean, how do you go now? Linear is there, one line only. But if you're trying now to do it differently, that there are billions of ways of doing this. So that has been a little bit of an issue, but there is no question that when you do this better sampling, then you will start even overlapping with the real pathway through some of these. And you have a much better chance to capture the real pathway. So in terms of what you do in this biasing, in all these biasing simulations, what you do is you have a biasing potential in addition to your force field potential. You introduce a biasing potential. It has a force constant, how stiff, how strong you are biasing the system. And then in a time-dependent manner, so this is the total simulation time that you can afford. And this is the time, every time step of your simulation. So you go from an initial state to a final state. So that's how much displacement change you want to have. in a particular time divided by t. That gives you in every step how much closer you want to get. And then you define this reaction coordinate. This is the property, the geometrical arrangement that you want to change in a time dependent. In 100 nanosecond, I want to go from a distance of 5 to a distance of 10. Or I want to go from an RMSD of 2 to an RMSD of 0. Or I want to go from an orientation of 5 degrees to an angle of 25 degrees. So this collective variable engaging a lot of atoms in the system. If it's helix, I just want to work with the C alpha atoms, for example, or the backbone atoms. RMSD, distance. For targeted MD, what is known in the field as targeted MD is RMSD. And it's still for simple transition, it's very powerful. If you have two domains just separating a little bit, RMSD works perfectly because it's a simple change and you move in the right direction. But we have radius of gyration, orientation quaternion, a little bit more advanced way of defining the position or orientation of an element independent of the reference, and other things. So they are all implemented in NAMD in COLVAR, collective variable, which is something I heavily recommend. And that's just as an example here. If you look at the work, how much effort it takes to induce a transition in 20 nanoseconds for a very complex protein. The red one is what you get when you use RMSD, collective variable. And in terms of the mechanism, how it goes, you can see we are going from the square to the triangle. These are the two end states. And in this particular case, I know that the intermediate is here. There is a third structure for this protein that is here. So if I do RMSD-based transition moving from here to there, it takes a mechanism. I'm analyzing it in terms of two angles, alpha and beta. It takes a mechanism that is incorrect. It takes too much work. The protein doesn't like this kind of transition, and the mechanism is wrong. Whereas if I start playing with my protocol and finding better protocols, lower work protocols, then I become also actually mechanistically correct in all these spaces that I can analyze the system. So I mean, now again, I repeat, doing multiple simulations is something you should always think about for a variety of reasons. Reproducing the results, one thing. You want to increase your sampling, it's much easier to run 5 simulations on supercomputers than one very long simulations. You can parallelize that better. You can submit 5 jobs in parallel. You want to sample how lipids are moving, or you want to have different arrangement of lipids around your protein. Instead of running one simulation and waiting for lipids to move, you can have 5 simulations with different initial arrangements, putting them in different parts of the configuration space initially, and then run 5 simulations each 20 nanoseconds. You get 100 nanosecond better sampling. You're worrying about the protonation state of your sidechain. So is it protonated, non-protonated? I mean, should I do months of calculation, quantum mechanics, or whatever? Or why not doing one simulation in protonated, one simulation in deprotonated form? Your ligand, it should be like this or should be like that. So just run 2 simulations. So don't forget, this is something that can be easily forgotten. Oh, sometimes it's much easier. Oh, let's try that one and this one. This is something that comes up a lot. So oftentimes, I mean, if you want to develop a new methodology, that's a different story. But if you have a very specific question that can be answered by multiple simulations, done. So anyway, so we do run on a lot of simulations, so we start actually seeing even mechanisms coming out of these. Alpha, beta, gamma are the different angles, orientations that we are using as collective variables for our biasing simulations. You can see beta is opening, closing of this end. Alpha is opening, closing of this end. For example, every time we change alpha before beta, the protein doesn't like it. It takes too much work. And the reason is that you are, before closing beta, you cannot open alpha. So it is mechanistically also wrong. And the protein mechanics the way these helices are arranged ensures that. That's very interesting. That's how this protein was selected by evolution to do this job, to be alternating access model, because its mechanic didn't allow both of alpha and beta to be open at the same time, which would violate the mechanism. So if you flip beta and alpha, everything becomes easier, and so on and so forth. So again, and based on this, so now we have sort of what we think is the complete structural transition cycle. It includes even the rotation of these two elements with respect to each other. We call it a doorknob. So you have to turn the doorknob before you open. Otherwise, it's high energy because the protein doesn't like it. So and the opening and closing of the two ends actually happen in two perpendicular planes. So these helices are like this, so they open and close like this here and like that here. So that's probably why they don't like to be open on both sides, because it's hard. So you have to close this, then it becomes easy to open it. So that's. And so another example here is that, again, I mean, now that was just the protein mechanics, so we did it also for another protein system in which now we want to see what is the effect of the substrate. So this is really, really hard because you're looking at a bias simulation, moving this beast using your collective variables, and you want to still be sensitive to whether you have a little substrate bound to the protein or not. If your pathway is wrong, if you're pushing it so hard through a wrong pathway, then you lose this sensitivity. One mutation or one substrate binding doesn't make any difference. So what we did was to break down our system. Let's say apo, inward facing, outward facing. So that's one leg of the thermodynamic cycle I would like to look at. So this is just the protein mechanics going from inward facing to outward facing. So that I do using the stuff I showed you for the other protein. Then for each of these states, they can bind the substrate. So I look at substrate binding in this leg, come from apo to bound. I look at substrate binding to outward facing state, come from apo to bound. And then for the bound state, what is the transition? So I'd like to know what's the free energy of this cycle. And here you can see, in addition to umbrella sampling that we talked about, we are using also what is called the replica exchange. That's another advanced methodology of enhanced sampling that allows you to go to a very large number of nodes because you have many, many replicas that are exchanging information with each other. So that's an advanced sampling technique. And then at the end, we put representations from this leg, this leg, this leg, and this leg to put together one single simulation with 150 replicas against this circular pathway to be able to calculate the free energy of the entire cycle here. So let me skip this. This is what we get. This is what was experimentally proposed, and this is not measurement. This is just a schematic saying, well, this is an antiporter. In the apo case, it should not be able to flip. The free energy of the transition between that inward facing, outward facing should be very high. Once the substrate binds, it should bring down the free energy so they can transition. Of course, the substrate binding itself creates a new state for each case, right? That's not going to be exactly the same. So that was a schematic, and it's amazing we are getting exactly that now with numbers, values, and error bars and everything. So this is really suggesting that the transition, the pathway, the mechanism, the collective variable, however, the reaction coordinate that we are using is not too bad. Otherwise, we wouldn't be sensitive to binding of a little phosphate to the protein. So you can see that in the apo case, the transition state has a very high barrier. In the bound form, upon binding, you create new states. Binding creates new state, OFB, IFB, and now the transition between them is much easier than that. So it's still very expensive, but I think this is, again, I think this is really something that people are investing a lot, new methodologies. So I have two other examples. So when is the lunch break? Oh, hi. Sorry. We are already in the lunch break, I guess, right?
I don't know, it could be 12:30, 20, or 12.
Oh, okay, okay. So then, okay. So good. So then, so now this, let me actually quickly just talk about it again, but because I think this bias simulations and non-equilibrium methodologies is something that we have to integrate. We are using them more and more. This is another example in which we are interested in membrane fusion, and this is a process that happens in neurotransmitter release. And here, defining a reaction coordinate is not trivial because there is no distance or radius of gyration, a simple thing to do that. So what we would like to be able to do is something like that. What would be the mechanism of membrane budding, for example, or the reverse of that would be the fusion that is facilitated by some proteins. So what kind of collective variable can you use to induce this process? And that's something that we thought we cannot be done so easily using standard techniques. So what we are doing here is, now what we are doing is to define, remember I told you about the external potentials that you can define as attractive potentials. So now I can design a shape transformation based on my favorite CAD program. You can design a series of shapes, and then I would like my system to follow this shape transformation by just converting it into potentials that are added as external potential. So here's an example, a simple parametric shape. So I have a sine function here, and I just add that to my flat membrane. It's a coarse grain simulation system, and I can induce a shape change. The molecular system follows. It's like SMD, right? SMD, if you pull correctly, nicely, then you can see something reasonable. If you pull too fast, you break the system. In fact, we had a lot of systems in which this couldn't follow we had to come up with a protocol. So now I can do it in a time-dependent manner. So now I have a series of potentials, and there is a smooth transition between them. So now you can see this is my next example of shape transformation. So that's just a waveform. This is a single simulation in which things are evolving according to what I tell it. And so where is my, okay, so goodness, the best example I had is not here. So, but let me go back to my, so, and I have actually this one is, ignore the left part. So this one, and I don't have this slide unfortunately, it's a series of shape transformation in which I am going from a combination of a flat and a spherical two objects, put them together, convert them to a mesh and potential, and you can see nicely I can form a vesicle. You can even trap some ions, could be neurotransmitters or other things. So these are, again, that sort of next generation of different kind of biases that we are putting into the system, sort of in terms of time-dependent shape transformation of biases. And finally, just one example of a reduced model. So, I mean, coarse graining is obviously a very common reduced model that is used to increase the time scale. But you can also come up with your own favorite kind of reduced representation. Here, for example, we were particularly interested in lipid diffusion, especially the head groups. There are a large number of proteins that are sensitive to the composition of the lipid composition of the membrane, lipid composition of the membrane. And in most cases, it's just the head group region that matters. So we argued that maybe we don't need to have all these long tails in the core of the membrane. We are mostly interested in the PS or PC or PIP2, all these negatively charged signaling lipids that interact with the protein. So we decided to remove that part, replace it with an organic solvent which has similar properties. We even created an in silico solvent that is liquid and hydrophobic and small. You can't have these three properties from anything in nature. It's either small and hydrophobic, but it's gaseous, or you can have only two properties out of three. So we created this model, but the point really is it's very powerful. If you are using computational techniques, you can come up with reduced representations just to focus on what you're interested in. So now we had, before this model, we had to pull this protein using SMD that we discussed, center of mass of this protein, pull it into the membrane and see how it binds. Of course, you don't know how far you have to pull this into the membrane, how strong it should be. And sometimes it pulls the membrane, it pushes the membrane down. It doesn't penetrate into the membrane. So the membrane binding process, which we wanted to simulate, wasn't even coming out of the simulations. So this was biased simulation, and we were not happy with the level of sampling we got. So what we did, using this reduced representation, which we call highly mobile membrane mimetic, awful acronym, HMMM, or model, that might be easier to remember for you guys, but we are stuck with that. So what happens if you look at lipids, this is a top view of a membrane. For a full representation, conventional membrane, this is how much diffusion you get in 10 nanoseconds for your individual lipids. Whereas if you go to the HMMM membrane, in 1 nanosecond, 10 times this shorter simulation, you start seeing even mixing of lipids. So lipid diffusion has been artificially enhanced without losing atomistic representation. It's not coarse grain. Coarse grain is also great, but here we were really interested in specific interactions of negative charges with these coagulation proteins, for example. So now no bias, we just place the protein in the membrane, in solution, and just watch it, how it interacts with these short-tailed lipids. This happens in 5 nanoseconds, so quickly. And because it happens very fast, now we can have proudly n equal 10 in our simulation system, and they bind, they all converge to a nice insertion depth and orientation. So you get a lot of sampling in terms of lipid-protein interaction, and now you can take one of these final models in which it's bound to the membrane, convert the lipids to a full membrane, and have your membrane-bound system. Otherwise, you have to place it somewhere deep, shallow. We don't know initially. If you place it initially, then you don't know what you're. And we have been applying it to a series of lot of different peripheral proteins that interact in a lipid-dependent manner to the surface of the membrane. We are also looking at insertion of other molecular species to the membrane. So I'd like to just end with one thing that is, again, another message I'd like to give is something that there are lots of approximations in simulations, and like any other methodology as we discuss, and we will see more of them tomorrow, actually. But it's very powerful in terms of actually testing hypotheses, and it's very powerful in terms of being playful, changing the conditions that cannot be done experimentally. So experimentally, you can mutate the sidechain. In simulations, you can also mutate the electrostatic of a backbone. I always say you can use a negative sodium ion in your simulation by just changing a little number in your simulation system. You can make a mass of something 10 times smaller or 10 times larger by changing a few numbers. So you can really design systems that test your hypothesis within the simulation system very nicely. So this is something that you need to keep in mind and take advantage of. That's a very powerful way, at least to show that within your simulation framework, the mechanism that your hypothesis is correct. You can test that there, and then hopefully it can be also tested experimentally. And this example I talked about, so we saw this change of the orientation of water molecules in the 2 half channels here. So we could have stopped there and said, okay, here we think there are helices and there are residues that cause this behavior, but instead of doing that, so we designed a couple of, this is our wild type behavior, if you wish. You have the flip of the dipole moment of water molecules in the 2 halves. And then we started mutating simulation, doing mutations in the system, first neutralizing some of the residues. In the NPA motif, it doesn't matter where they are. Then we turned off electrostatics of these two half helices to test whether they can affect the wild type behavior. And when we combine electrostatics, we're turning off the NPAs and the two helices, then you get the relaxed structure of water. So by adding this to the paper, we showed what elements in the protein are important for this very peculiar behavior. So that's very powerful, and keep that in mind. Okay, so I just end with this one. This was a picture actually we did together with Klaus. This was I was still a postdoc with Klaus when we did this paper, and this was a very popular representation of how water orientation flips from one side to the other. Okay, so I asked everybody to ask 10 questions, Yvette. If you are too late. So let's see. Questions? Yes?
When you have multiple transition pathways for the refined QwikMD, do you have multiple collective variables or collective variables which are specific to each pathway?
Yeah.
Normally
So,
aren't.
right. So I mean, I wouldn't call them TMD. TMD is RMSD based. That's the standard definition. So let's stick with that. So everything else, we are using different protocols. And the protocol is a combination of collective variables, force constant, and the sequence of collective variables. Sometimes you have, let's say, alpha and beta, opening and closing of the two ends. Should we do them together? Should we do alpha first, beta next, beta first, alpha next? We call them different protocols, right? Each one of them gives you a different mechanism, different pathway, different type of transition. It could be simply force constant. Maybe this one has a high force constant, this one has a lower force constant. That's a different protocol. So we do these things. We want to make sure that our protocol creates our end state. That's number one criterion. So you should go there. If it doesn't take you there, it's of no use. But then once you're there, how much work, how much effort it took using those manipulations to go there. So then the first round you get some idea, oh, this is too hard, this is 500 kilocalorie per mole higher, forget about that. You get some idea, then you have to repeat them because these include a lot of dissipative component in them. We repeat some of them, we focus a little bit more on some of them. It's by no means comprehensive, as we discussed, but it gets you actually, every time we move away from RMSD, we get much easier ways or more compliant protein kind of transitions. Yeah? Yes?
So you were talking at the end of it about mutations, like having your wild type and having the mutations, and doing the simulations for them. Are you able to do the mutations of a system that you have already run a simulation, or do you normally do the mutations in the building component before you run the simulation?
We usually start from the beginning. So because again, you don't know what happened during your wild type simulations. I mean, it's easier to write the method section if you say, I started with the PDB, one wild type, and I did the mutation there and continue there. But I guess I don't see any problem if you feel for some reason, oh, you want to know a particular state that you sampled in your simulation, the second basin. You want to mutate that one as well. You don't want to wait another, 100 nanoseconds to reach there. Maybe you can have two mutation simulation, one with the initial PDB, one with that new state that you discovered, and see whether it brings it back or does something else. You can also think about scenarios in which you are interested in mutating one of the frames from your wild type trajectory. That's fine. Yeah. So we have done, for example, lipid packing takes time. For example, maybe you have spent a lot of time getting a nice packing of your lipids around the protein, and now you just want to mutate a residue inside the lumen, so it doesn't make sense to go back and repeat this microsecond. You keep the lipid shell and everything, you just do the mutation in your equilibrated system. That's fine. 8 more, yes?
I have two questions. The first one is about
Why you need to minimize?
Yeah, minimize heating and equilibrium, and why we do the theory.
I mean, you do minimization to remove very high force regions or hot spots that might come from your crystal structure transitioning to your force. I mean, nowadays, all the structures are kind of minimized by some force field, so it's not too bad. But a long time ago, some structures were very bad. You know, two close atoms, they would crash your simulation. You have to minimize to remove these hot spots, so to say. Is that clear? Well, then when you minimize that, then at some point, you have to introduce the temperature. You have to bring kinetic energy to the system, right? So that's the thermalization. And if your system has issues, then you want to do it slowly, maybe a stepwise. Sometimes you don't even have to minimize the PDBs we have these days because they are so good and nice and relaxed. You just go and they work. If it works, it's fine. But sometimes you get stuck, and you have to minimize them a lot. You have to start with even shorter time steps to slowly let them move. You know, you have to do a lot of tricks. In the beginning, the first picosecond can be very tricky. The equilibration is just part of the production run. You have to let the system to sample to reach an equilibrium ensemble, right? That's part of your production kind of things. So.
My second question is on the size of the water box. So directly we want to minimize the size of the water box because we want to put additional computational to perform the additional size.
Mm-hmm.
I may wonder how do we determine the size of water box for the system?
Well, I mean, the size of the system, as I tried to point out, is not too much of a problem. So again, I mean, larger systems scale much better with a larger node count. So that's usually not a major issue. So yes, you don't want to make the system too big. So what is the minimum size? Again, I think one simple rule is that you don't want the copies. You have a periodic boundary condition system, right? You don't want the copies of the same protein to see each other. So your cutoff distance is 10, 12 angstroms. So you want to make sure that there is at least 12 angstrom distance between the two proteins that are sitting in the middle, at least. So that's a simple rule, I would say. If you have a strong dipole or strong charges somewhere in one end of your protein, that becomes more important to have more water padding. But if you want me to give you a number, I would go with the cutoff distance. You don't want the copies to be closer than 12 angstrom because then they start seeing each other in the direct part of the electrostatic. Yeah.
Thank you. This has been great, but we have a photographer who
Oh, right.
a group picture.
Okay.
Can a group picture be taken